You have been awake for hours. Maybe. And sleep came. But this morning you felt it anyway. That tired, that sleep did not fix. Your body logged the hours. But your brain may not have gone where the hours required. There is a stage of sleep so deep that most people spend less than 20 minutes there each night. And almost no one knows they are missing it. Welcome to Sleepy Mind Science. Tonight begins at the bottom of sleep, where the brain does what waking never could. Close your eyes and breathe slowly. Let the day release its grip. You are already moving toward the place these facts describe. And now we begin. Sleep is not one thing. It is a cycle. And inside that cycle, there is a stage that scientists call N3, or slow wave sleep. This is the deepest level of non-REM sleep, where your brain shifts from the fast, irregular patterns of waking into something far slower and more synchronized. The brainwaves in N3 are called delta waves. They move at less than 4 cycles per second, and they are among the largest, most powerful electrical patterns the brain produces. When researchers ask what your brain is actually doing in the deepest hour of sleep, this is where the answer begins. Not in dreaming, not in the light drift between waking and rest. In the delta, in the slow wave. The word slow in slow wave sleep is precise. Delta waves are not slow because the brain is less active. They are slow because enormous numbers of neurons are firing together in a long, coordinated wave, then going quiet, then firing again. This alternation, called the up state and the down state, happens roughly once per second during deep sleep. During the up state, neurons are briefly as active as they are in waking. During the down state, they go nearly silent. What looks like the brain powering down is actually the brain cycling, building, releasing, building again. A sleeping brain is not an idle brain. It is a brain running a protocol it cannot run while you are awake. You enter N3, sleep roughly 30 minutes after falling asleep. Before you reach it, you pass through 2 lighter stages. N1, a brief threshold state where you may still half hear the room, and N2, where your body temperature drops and your heart rate slows. N2 is where most people spend the most total time in any given night. But N3 is different in quality, not just depth. It requires the brain to build enough sleep pressure, a biological drive called adenosine, before it can shift into delta. If you went to bed later than your body wanted, or if you woke once already, that pressure is still available. The brain will try to use it. The first deep sleep episode of the night is usually the longest and most intense. The sleep pressure that drives you into N3 is a molecule called adenosine. Your brain produces adenosine throughout every waking hour as a byproduct of neural activity. The longer you are awake, the more adenosine accumulates in the spaces between neurons, and the more urgently the body signals the need for sleep. Caffeine works by blocking adenosine receptors, which is why it delays fatigue without actually reducing the underlying accumulation. When you finally sleep, the brain clears adenosine, slowly, specifically. Over the course of the night, the first clearing surge happens during N3. This is part of why the deepest sleep tends to occur in the first third of the night, and why cutting sleep short leaves a residue that a nap does not fully address. The brain does not choose to enter deep sleep. It is pulled in by biology. Two separate systems regulate this: the circadian clock, which tracks time of day cues like light and darkness, and the homeostatic sleep drive, which tracks how long you have been awake. These two systems normally align. The circadian clock signals maximum sleep opportunity around the same time the homeostatic pressure peaks. When they fall out of sync, through irregular schedules, shift work, or late-night light exposure, the brain may have inadequate sleep pressure at the moment the circadian window opens, and N3 is the first casualty. You may lie down at the right time and still not reach the bottom of sleep, because the conditions that open the door to slow-wave rest are specific. N3 sleep is most concentrated in the first half of the night. In the second half, the brain shifts its balance toward REM sleep, the dreaming stage, associated with memory integration and emotional processing. This is not random. The brain appears to sequence restorative stages, purposefully deep, physical and cellular repair first, then cognitive and emotional consolidation. People who go to bed at midnight instead of 10 and wake at 7 are still sleeping 8 hours. But they have disproportionately less N3 and more of the late-night REM that fills the final sleep cycles. The total duration looks the same. The internal composition has shifted. This is one reason researchers now focus not only on sleep duration, but sleep architecture, the proportion and timing of each stage. During N3 sleep, your body releases the majority of its nightly growth hormone. This hormone does not only govern childhood development. In adults, it is a primary signal for cellular repair, directing the immune system, rebuilding muscle tissue, consolidating bone density, and stimulating the production of proteins that maintain organ function. The release is timed to the slow wave period, the first and longest N3 episode, roughly 45 to 90 minutes after sleep onset. Triggers the largest single surge of growth hormone in any 24-hour period. Disrupting this window through alcohol, late eating, artificial light exposure, or simply sleeping too late, compresses or eliminates the N3 episode and blunts the hormone release. The body still functions, but the repair brief was shorter than it needed to be. The immune system is regulated, in part, by sleep. T cells, white blood cells that identify and destroy infected or abnormal cells, become significantly more adhesive during deep sleep. This is not a general immune boost. It is a specific molecular change. Hormones called prostaglandins, which promote inflammation and reduce T-cell stickiness during waking, fall sharply during N3 sleep. The T-cells then gain access to surfaces they cannot reach when prostaglandins are elevated. Researchers have documented this mechanism in detail. The window of enhanced immune function is open during slow-wave sleep and closes with waking. Over many nights of shortened or fragmented deep sleep, the aggregate effect on immune competence becomes measurable. Your brain has a drainage system that operates almost exclusively during sleep. It is called the glymphatic system, named by combining glial cells with the lymphatic system it resembles in function. During waking, cells in the brain are packed relatively tightly, and cerebrospinal fluid moves slowly through the narrow spaces between them. During deep sleep, these glial cells shrink noticeably, expanding the interstitial space and allowing cerebrospinal fluid to flow through at a much faster rate. This surge washes out metabolic waste products, including proteins associated with neurological disease. The mechanism was first described in mice in 2013 and human evidence for the same process continues to accumulate. Scientists treat the basic clearance function as established. The precise clinical implications remain an active area of study. One of the waste products cleared during deep sleep is beta-amyloid, a protein that accumulates in the brains of people with Alzheimer's disease. The connection between sleep and neurological health is still being established, and causation is difficult to separate from correlation. What the evidence supports, beta-amyloid clearance increases during N3 sleep in both animal models and human imaging studies. And people with chronic short sleep show higher rates of amyloid accumulation over time. Whether better sleep prevents disease, or whether early disease disrupts sleep first, remains an open question. But the relationship between deep sleep and what the brain holds onto, and what it releases, is now one of the most active areas in sleep neuroscience. Memory consolidation, converting short-term experiences into durable long-term memories, depends on both REM and non-REM sleep, but the contributions are different. During N3 sleep, the hippocampus replays the events of the day in compressed, accelerated sequences, and transfers the encoded information to the neocortex for long-term storage. This transfer is called systems consolidation. It does not require conscious awareness. The hippocampus is working while you are deeply unaware of anything. Research using electroencephalograms has captured the signature of this process. Sharp wave ripples from the hippocampus appear synchronized with the slow oscillations of deep sleep, as if one system is timing its transmissions to the other's rhythm. The slow oscillations of N3 sleep appear to act as a coordinating clock for memory transfer. Each down-to-up transition in the delta wave may serve as a trigger, the moment when hippocampal ripples most effectively reach the neocortex. In studies where slow-wave oscillations were artificially enhanced during deep sleep, through gentle auditory tones timed to the brain's own rhythm, participants showed better performance on memory tasks the following morning, compared to participants who slept the same amount without enhancement. The brain, under the right conditions, was able to use more of its own repair window. This is one of the more intriguing findings in current sleep science. The process is improvable, not fixed. The neocortex, the outer layer of the brain responsible for language, reasoning, and conscious thought, plays a receiving role during deep sleep. While the hippocampus replays and transmits, the neocortex integrates. Slow wave oscillations travel across the neocortex in coordinated sweeps, appearing first in the frontal regions and moving backward toward the temporal lobes, roughly once per second during each delta cycle. This traveling wave ensures that different parts of the cortex receive updated information in sequence. The architecture of N3 sleep is not random. It has a directed topology, a shape, and the shape is built for transfer. Motor learning, the acquisition of physical skills, from typing to instrument playing to sports technique, is particularly sensitive to N3 sleep. Researchers have found that people who learn a new motor sequence and then sleep show greater overnight improvement than people who practice the same sequence and stay awake. The improvement is not in performance during practice. It emerges the next morning, after sleep has processed what the waking brain could not. And when N3 sleep is specifically disrupted, while REM sleep is left intact, the overnight improvement disappears. The muscle did not learn. The brain did not consolidate. The movement was practiced but not retained in the way sleep would have retained it. The The brain does not simply record memories during N3 sleep. It edits them. Emotional charge is stripped from declarative memories during deep sleep, particularly from memories that arrived with high stress or anxiety. The process involves the prefrontal cortex gradually gaining access to hippocampal memory traces and modulating the emotional tagging associated with them. This is part of why the same event, recalled the morning after a full night of sleep, often feels less urgent, less raw, than it did the night before. The facts are retained. The alarm attached to them is reduced. Sleep is not forgetting. It is filing, placing what happened somewhere that no longer requires the same vigilance to hold. Pain sensitivity increases with sleep deprivation, and specifically with reductions in deep sleep. N3 sleep appears to have an analgesic function. The body's pain threshold rises during this stage, and the nervous system recalibrates its baseline sensitivity overnight. Studies that selectively disrupted slow-wave sleep in participants, using quiet tones that prevented deep sleep without waking subjects fully, found that participants reported increased pain sensitivity the following day, even with normal total sleep duration. This mechanism may explain, in part, why chronic pain conditions are frequently associated with disordered sleep, and why the relationship between pain and sleep tends to be bidirectional. Each disrupts the other. Core body temperature drops to its lowest point of the night during deep sleep. This is not incidental. Falling body temperature is one of the primary biological signals that initiates the shift into N3. The hypothalamus, the brain region that regulates both sleep and temperature, begins cooling the body as darkness approaches, partly through skin blood vessel dilation, which releases heat from the extremities. A cool bedroom accelerates this process. A warm one delays it. The core temperature minimum during deep sleep reached somewhere between 2 and 5 in the morning for most people, also coincides with the period of lowest alertness and slowest reaction time. A vulnerability that exists precisely because the brain is using that window to do something that cannot wait. The basal ganglia, a cluster of structures deep in the brain involved in movement, reward, and habit formation, shows a distinct activity pattern during N3 sleep. It does not simply go quiet. It continues firing, but in ways that appear linked to the consolidation of procedural and habitual behaviors. This is separate from the hippocampal neocortical transfer of declarative memory. The brain runs two memory consolidation processes in parallel during deep sleep, one for explicit facts and events, one for automatic habits and skills. When you wake from deep sleep and a formerly difficult movement suddenly feels easier. This is why. The basal ganglia worked overnight without any instruction to do so. Cortisol, the stress hormone, reaches its lowest point of the 24-hour cycle during the first half of the night, precisely coinciding with the peak N3 window. This is not coincidence. The hypothalamic pituitary adrenal axis, which controls cortisol release, is suppressed during slow-wave sleep by the same neural signals that deepen sleep. Cortisol begins rising again in the early morning hours, producing the gentle physiological arousal that prepares the body to wake. People with chronic insomnia or sleep fragmentation show elevated cortisol during the first part of the night, suggesting the suppression is incomplete. This keeps the stress response partially active during the hours when the brain most needs it quiet. Heart rate variability, the variation in time between heartbeats, used as an index of autonomic nervous system balance, increases during N3 sleep. Higher heart rate variability is associated with greater parasympathetic dominance, meaning the rest and repair branch of the nervous system is in control. During deep sleep, the sympathetic nervous system, which governs the fight or flight response, is substantially suppressed. Blood pressure falls. Arterial walls relax. The cardiovascular system undergoes its own form of rest. Not inactivity, but restoration. Chronic reductions in N3 sleep are associated over time with increased cardiovascular disease risk, hypertension, and elevated resting heart rate. Consequences that accumulate across years of shortened or disrupted deep sleep. The thalamus, a relay station in the center of the brain that routes sensory information to the cortex, plays a central role in protecting deep sleep. During N3, the thalamus generates bursts of electrical activity called sleep spindles, which act as gates. They briefly and rhythmically suppress the thalamic relay, preventing external sensory signals from reaching the cortex and disturbing the ongoing slow-wave activity. This is how you can sleep through background noise that would normally register as alerting the thalamus is actively filtering. Sleep spindles are visible on EEG recordings as brief bursts of fast activity overlaid on the slower delta waves. Their frequency and quality vary by age, genetics, and mental health, and they are associated with cognitive flexibility and the ability to retain learned information overnight. People who consume alcohol before sleep often report sleeping well and deeply. But the neurological picture is different from what the experience suggests. Alcohol accelerates the transition to sleep and increases N3 duration in the early part of the night, which is why it produces heavy early night sleep. But it disrupts the architecture of the second half. REM sleep is suppressed, and N3 rebounds abnormally later in the night, fragmenting the cycle and disrupting the coordinated sequence the brain uses for repair and memory consolidation. The subjective sense of deep sleep is produced. The functional output, the consolidation, the clearance, the hormonal cascade, is not fully completed. The sleep alcohol provides is a structural approximation of deep rest that does not deliver the same result. N3 sleep decreases steadily with age, beginning in early adulthood. A young adult in their 20s may spend 20 to 25% of their total sleep time in slow-wave sleep. By their mid-40s, that percentage has typically fallen to 10 to 15%. By their mid-60s, some individuals spend nearly no measurable time in N3. The brain does not abandon the attempt. Delta waves still appear, but their amplitude diminishes. And the coordinated synchrony that defines productive slow-wave sleep degrades. This age-related decline is one reason cognitive sharpness tends to erode over decades even in otherwise healthy adults. The brain's nightly maintenance window gets shorter, not because the tasks are smaller, but because the conditions for doing them become harder to sustain. Exercise has one of the most consistent documented effects on slow-wave sleep. People who exercise regularly, particularly aerobic exercise, spend more time in N3, show higher amplitude delta waves, and report greater sleep depth and morning restoration than sedentary individuals. The mechanism involves several pathways. Exercise increases adenosine accumulation, raising sleep pressure. It temporarily elevates core body temperature, which then drops more steeply in the evening. And it modulates growth hormone sensitivity. making the hormonal cascade of N3 more effective. The timing of exercise matters. Morning or afternoon exercise reliably improves slow-wave sleep. Exercise within 2 to 3 hours of bedtime can delay sleep onset, though this varies considerably across individuals. 25 facts in. A pattern is visible. The brain does not wait for permission to repair. It runs a highly sequenced protocol the moment you reach deep enough sleep. Clearing waste, consolidating memory, releasing growth hormone, suppressing cortisol, recalibrating pain sensitivity, and regulating cardiovascular function, all within the same coordinated window. The central question, what your brain is actually doing in the deepest hour of sleep, is beginning to have an answer. But it opens another layer. What happens when that window is interrupted before the protocol completes, when the repair runs, but not long enough? That is where the next arc begins. Right now, there is slow-wave activity in your brain, not because this fact told it to be there, because you have been still long enough. The adenosine that built through your day has been clearing since you lay down. The thalamus has been filtering what it does not want to reach you. The question that just opened, about what happens when the repair window closes too early, lives in your own chemistry, in the difference between the night that finished the work and the one that nearly did. Notice your breath. The delta already knows what to do with it. Now we continue. Sleep fragmentation, waking during the night, or moving out of deep sleep before completing a full N3 cycle, is more common than most people realize, and its effects are often attributed to other causes. Stress, afternoon fatigue, difficulty concentrating, emotional reactivity. These are frequently experienced as separate problems, but research consistently links them to disrupted sleep architecture, particularly the loss of N3. Brief arousals, lasting only seconds, occur dozens of times each night without producing full waking. Each one interrupts a slow-wave cycle that was either building or recovering. The brain logs the disturbance. The repair continues, but from the beginning of the cycle, not where it left off. Sleep apnea is one of the most common causes of slow-wave sleep disruption worldwide, and one of the most underdiagnosed. In obstructive sleep apnea, the airway partially or fully collapses during sleep, reducing oxygen levels and triggering a brief arousal to restore breathing. These arousals often do not wake the person fully. The sleeper may have no memory of them, but they reliably pull the brain out of N3. A person with untreated moderate sleep apnea may be spending 8 hours in bed while their brain cycles repeatedly out of deep sleep every 5 to 10 minutes. They wake feeling as though they never reached the bottom of rest, because, neurologically, they have not. The relationship between deep sleep and mental health is bidirectional and specific. Reduced N3 sleep is associated with increased symptoms of depression, anxiety, and post-traumatic stress. But causation runs both ways. Depression reduces slow-wave sleep amplitude. Reduced slow-wave sleep worsens mood regulation. Anxiety elevates nighttime cortisol, which suppresses N3. Suppressed N3 leaves the emotional content of the day less processed by morning, increasing baseline anxiety. The system is looped. Treatments that directly target sleep architecture are showing promising outcomes in clinical trials. Improving deep sleep in people with mild anxiety or depression often produces measurable mood improvements within 1 to 2 weeks. Slow-wave sleep and the default mode network. The brain system active during mind wandering, self-reflection, and imagination have a precise relationship. During waking, the default mode network is one of the most metabolically active regions of the brain. During N3 sleep, it goes quiet. This matters because the default mode network is also the primary generator of the rumination and self-referential thinking that prevents sleep in anxious individuals. The shift into deep sleep is, among other things, a shift out of self-focus. The brain stops narrating your experience and begins processing it. This is one of the reasons deep sleep feels unlike anything else in the 24-hour cycle. It is the only state in which the self-monitoring system fully powers down. The prefrontal cortex, the brain region responsible for decision-making, impulse control, and complex social behavior, is among the areas most sensitive to N3 sleep reduction. A single night with reduced slow-wave sleep measurably impairs prefrontal function the following day. Working memory decreases. Emotional reactivity increases. The ability to hold multiple pieces of information in mind simultaneously weakens. These effects are modest after one night. They compound after multiple nights. Chronic partial sleep deprivation, 6 or 6.5 hours instead of 8, night after night, produces prefrontal deficits that people consistently underestimate. Studies show the same pattern. People feel they have adapted to less sleep. Their performance data disagrees. Thermal regulation during deep sleep is active, not passive. The body does not simply cool because activity has slowed. Specific neurons in the hypothalamus, called warm sensitive neurons, increase their firing rate when they detect that skin temperature is rising, and they actively orchestrate the heat dissipation that pulls core temperature down. This process requires good circulation and responsive peripheral blood vessels. Age-related changes in thermoregulation, particularly reduced ability to dilate peripheral vessels, are one reason older adults have more difficulty entering deep sleep. Cool bedroom environments assist by accelerating the external temperature gradient that the hypothalamus is trying to create internally. The body and the room are working together toward the same temperature destination. Insulin sensitivity. The body's ability to respond appropriately to glucose improves during N3 sleep. Slow-wave sleep is associated with lower sympathetic nervous system activity and reduced cortisol, both of which impair insulin signaling when elevated. Studies in which healthy young adults were subjected to 3 nights of selective slow-wave sleep disruption showed significant reduction in insulin sensitivity, comparable in magnitude to the effect of gaining significant weight or aging a decade. The metabolic function of deep sleep is not trivial. It is a recalibration of the body's glucose management system that occurs each night, resets with each N3 episode, and accumulates in its absence. The ghrelin and leptin system, hormones that regulate hunger and satiety, is disrupted by short sleep, with specific sensitivity to N3 reduction. Ghrelin, the hunger hormone, rise. Leptin, the satiety hormone, falls. This shift appears within a single night of insufficient deep sleep. The brain is not responding to actual caloric deficit. It is responding to the signal of inadequate restoration. In a modern environment with constant food access, this mechanism produces next-day overconsumption, particularly of high-calorie foods, that is difficult to override through conscious intention because it is driven by hormones, not hunger in the familiar sense. The relationship between deep sleep and learning is not uniform across all types of knowledge. Factual, declarative memories, names, events, explicit information rely heavily on the hippocampal neocortical transfer of N3, but emotional memories are processed differently. Emotional learning, the association of a cue with a fear, a reward, or a social consequence, is consolidated partly during REM sleep, which involves different circuitry. The brain handles different kinds of knowing in different sleep stages. A full night with intact sleep architecture is not redundant. Each stage is completing a different file. When a stage is skipped, specific categories of learning are selectively impaired. The spine of the vertebrate body, including the spinal cord and peripheral nervous system, undergoes maintenance during sleep that is distinct from brain activity. Glymphatic-like clearance has been identified in spinal tissue. Cerebrospinal fluid that flows through the brain continues into the spinal canal, carrying waste products from neural metabolism. During deep sleep, when brain fluid dynamics are most active, this spinal clearance also increases. Chronic disruption of deep sleep may thus impair maintenance across the entire central nervous system. Not only the brain regions that are most studied, but the full length of the neural architecture that runs the body. The brain's reward system, centered on dopamine pathways in the ventral striatum, is directly regulated by deep sleep. N3 sleep appears to reset dopamine sensitivity, allowing the reward system to respond normally to moderate pleasures the following day. Sleep deprivation, and specifically slow-wave sleep reduction, has been shown to blunt the response to positive stimuli while amplifying the response to negative ones. The world feels flatter after poor deep sleep, less pleasurable, less interesting, while frustrations feel sharper. This is not a mood problem in the familiar sense. It is a neurochemical shift produced by incomplete restoration of the reward circuitry that deep sleep maintains. The cerebellum, a structure at the back of the brain involved in coordination, balance, and the fine-tuning of movement, is highly active during slow-wave sleep. Cerebellar activity during N.3 is associated with the consolidation of movement sequences learned during the day. This is the mechanism by which sleep transforms effortful, consciously controlled movements into fluid, automatic ones. Musicians, athletes, and surgeons who prioritize sleep during training periods show faster skill acquisition than those who reduce sleep to increase practice time. The brain needs the sleep to process what the body practiced. Practice without subsequent deep sleep produces a different kind of learning. Shallower, less automatic, more fragile under stress. N3 sleep architecture varies between individuals in ways that are partly genetic. The amplitude of delta waves, how tall and synchronized the slow oscillations are, is significantly heritable. Twin studies show that identical twins have more similar slow-wave sleep patterns than fraternal twins. Some individuals are, by genetic disposition, deeper sleepers with more productive N3 windows, while others have shallower delta waves, regardless of their behaviors. Behaviors still matter. Sleep hygiene, light exposure, exercise, and timing all modulate N3 depth. but the ceiling and the floor are partly set by inheritance. This explains why people respond differently to the same amount of sleep deprivation. The length of an individual N3 episode is not fixed across the night. The first episode, occurring roughly 40 to 90 minutes after sleep onset, is typically the longest, lasting anywhere from 20 to 40 minutes. Subsequent N3 episodes, in the second and third sleep cycles, are progressively shorter. The homeostatic sleep pressure is highest at sleep onset and falls as the night progresses. The brain uses the high-pressure window to do its most intensive slow-wave work first. This is why the first few hours of sleep are disproportionately restorative. Sleeping from midnight to 7 is not equivalent to sleeping from 10 to 5, even at the same duration. Artificial light at night, particularly blue spectrum light from screens, delays the circadian signal that initiates sleep, which in turn delays the onset of N3. Specialized cells in the retina respond to blue light by sending signals to the suprachiasmatic nucleus, the brain's master clock, that suppress melatonin production. Melatonin is not a sleep drug. It is a timing signal. When it is delayed, the biological evening that precedes deep sleep is pushed back, compressing the N3 window even if total sleep duration remains unchanged. The body still gets 8 hours, but the first episode of deep sleep begins later, and the architecture of the night shifts. You've been listening for a while now, thoughts still quieting like a room after the last conversation ends. Just notice where you are, whether you are still mostly here or already somewhere softer. We're moving through the science of the brain's deepest hours together, the part most people are asleep for, without knowing what it is doing. Let's continue, slowly. The relationship between deep sleep and the immune response to vaccination is now well documented. Studies examining participants who received influenza vaccines found that those who slept normally in the nights following vaccination produced significantly higher antibody titers than those who were sleep-deprived. The immune system requires slow-wave sleep to consolidate immunological memory in a way that parallels how the brain consolidates learned information. The T cells activated during vaccination are processed and filed during N3. A reduced deep sleep window means a less efficient immunological learning period. This has direct implications for vaccine efficacy, a finding that has begun to influence recommendations around sleep before and after vaccination. The adenosine clearance that occurs during N3 sleep is not a continuous trickle. It appears linked to the up-down oscillations of the slow wave itself. During the down state, the brief silence between firing bursts, certain channels in glial cells open and allow adenosine to be reabsorbed or broken down. The tempo of clearance is matched to the tempo of the delta wave. A brain with lower amplitude or less synchronized slow wave activity clears adenosine more slowly, accumulates more residual sleep pressure, and is more likely to produce persistent grogginess in the morning. The biology of feeling rested is not about how long you slept. It is about how cleanly the slow wave ran. Children, compared to adults, spend a dramatically higher proportion of sleep time in N3. Infants may spend 50% or more of their total sleep in slow wave stages. Children between the ages of 5 and 10 typically show the highest amplitude delta waves of any age group. The slow oscillations are large, synchronized, and long. It is because the brain's developmental program requires deep sleep as active infrastructure. The connections being formed between neurons during waking The synapses strengthened through play, language, and experience are pruned and stabilized during N3 sleep through a process called synaptic homeostasis. The brain does not grow by adding everything. It grows by deciding what to keep. Synaptic homeostasis theory, proposed by neuroscientists Giulio Tononi and Chiara Cirelli, holds that during waking, the brain strengthens connections through learning and experience, gradually increasing the overall synaptic weight of the neural network. If this continued unchecked, synaptic connections would become so dense that the signal-to-noise ratio would collapse. Deep sleep renews the system by downscaling synapses, not eliminating them, but reducing their baseline strength in a way that preserves the patterns learned, while restoring the network's capacity to learn again the next day. Each morning, the brain wakes up with a selectively pruned network, quieter, more sensitive, more ready. The night did not rest the brain. It refreshed it. Sleepwalking and sleep terrors, both parasomnias or abnormal behaviors during sleep, occur specifically during N3, not during dreaming, not during light sleep, but in slow-wave sleep. This tells us something about the nature of deep sleep. It is a state of profound motor and cognitive disconnection from waking, but not a state of complete shutdown. The brain is active during N3 in ways that can produce complex, partially coordinated behaviors. Walking, eating, speaking, without producing memory or consciousness. People who sleepwalk have no recollection of it. From the inside, deep sleep is felt as nothing. From the outside, it is the brain running processes that have no place for an observer. The concept of sleep inertia, the grogginess experienced immediately upon waking, is most severe when waking occurs during N3 sleep. Forced awakenings from deep sleep produce cognitive impairment that can last from 15 minutes to 2 hours. The slow-wave delta state is so deeply suppressive of the arousal systems that transitioning to full waking takes time. The brain must shift from coordinated, synchronized delta mode to the fragmented, fast-frequency patterns of alert waking, and the transition is not instantaneous. This is why alarms are cognitively disruptive in a way that natural waking is not. Natural waking tends to occur from light sleep, or REM. At the end of a 90-minute cycle, alarm-forced waking can terminate any stage, including N3. Meditation practice, specifically mindfulness-based practice with consistent long-term use, has been associated with changes in sleep architecture, including increased N3 duration and amplitude. The mechanism proposed is plausible. Meditation reduces activity in the default mode network during waking, lowers baseline cortisol, and reduces evening ruminative thinking, the exact factors that interfere with the transition to deep sleep. Long-term meditators show different slow-wave patterns from non-meditators, even on nights when they do not meditate before sleep. The practice appears to change the brain's baseline regulation in ways that carry into the night. This is not a universal finding, and effect sizes vary across studies, but the direction of the evidence is consistent. The architecture of slow-wave sleep is influenced by the emotional content of what happened during the day. Experiences with high emotional salience, particularly those involving stress, social conflict, or significant achievement, appear to elevate the intensity of subsequent N3 slow waves. The brain seems to prioritize processing emotionally charged material during deep sleep. allocating more slow-wave resources to the integration of significant events. This may be why nights following emotionally intense days sometimes feel heavier. A quality of depth that is real, not imagined. The brain is doing more work, not less, on those nights. Temperature manipulation is one of the most reliable and accessible tools for increasing slow-wave sleep. A warm shower or bath taken 90 minutes before bed, not immediately before, produces a transient rise in skin temperature, followed by a sharper fall as the body dissipates heat through dilated peripheral blood vessels. This fall in skin temperature accelerates the internal temperature drop that initiates deep sleep. Studies have measured the effect. A 90-minute advance warm bath reliably increases N3 duration by a small but consistent amount. The mechanism is the same one the body uses naturally. The bath creates a more pronounced temperature gradient, and the brain uses it. Halfway through the facts of tonight, the question that opened these hours has a partial answer. The brain, in its deepest sleep, is doing something irreplaceable. A coordinated repair protocol that cannot run while you are awake, cannot run well in light sleep, and cannot be fully postponed without cost. But the question the second half follows is more personal. How does the brain know whether the protocol ran, and what changes when it does not? Not after one night, but gradually, quietly. Across weeks and months of incomplete deep sleep, the cost is not obvious in the morning. It accumulates in the afternoon, in the year, in the years. That is where tonight continues. What just opened? The question of what accumulates when the deep sleep protocol runs short night after night is not only a neuroscience question. You may already know the answer in your body. The tired that arrived after a stretch of months when sleep was technically there but never quite enough. That's not exhaustion from not enough hours. That's something the second half of tonight traces. Feel the weight of where you are lying. The stillness around you now is the same stillness the brain is using. The repair is already underway, whether you stay awake for it or not. Now we continue. The cumulative effects of chronic slow-wave sleep reduction are visible in long-term health data. Large epidemiological studies, following tens of thousands of participants over years, have found that people who consistently sleep 6 hours or less, or who show disrupted sleep architecture on objective measurement, have higher rates of cardiovascular disease, type 2 diabetes, metabolic syndrome, and cognitive decline compared to those sleeping 7 to 9 hours with intact slow-wave stages. The effect persists after controlling for weight, physical activity, diet, and stress. The nightly repair window is doing something that no other interval in the 24-hour cycle replicates. The delta waves of deep sleep have a precise spatial organization. In young adults, the highest amplitude slow oscillations originate in the frontal lobes and sweep backward toward the occipital and temporal cortex. This topography is not random. The frontal cortex is the region most active during complex cognitive work, and the region that accumulates the most synaptic strengthening over the course of a day. The slow wave starts where the debt is largest, and moves from there. In older adults, the amplitude of frontal delta declines first and most steeply, a neurological signature that precedes measurable cognitive aging by years. Napping can partially restore some functions that depend on slow-wave sleep, but with constraints. A nap of 20 to 30 minutes typically contains N1 and N2 sleep, but rarely reaches N3. a 60 to 90-minute nap may contain a brief N3 episode, enough to produce some growth hormone release and partial adenosine clearance. But it cannot replicate the sequenced architecture of a full night, which places N3 first and most intensely. Late-day naps can also reduce the homeostatic sleep pressure that drives the following night's N3, making the deep sleep window shallower. A nap addresses the fatigue. It does not complete the protocol. The thalamic spindle activity that protects deep sleep is also involved in the transfer of memories from the hippocampus to the cortex. Sleep spindles. Brief bursts of fast oscillatory activity are precisely timed to coincide with the upstate of each slow oscillation and with sharp wave ripples from the hippocampus. The three events, the slow wave, the spindle, and the hippocampal ripple, occur in a precise sequence that researchers call the slow oscillation spindle ripple coupling. When this coupling is intact, memory consolidation is efficient. When it is disrupted by sleep disorders, by certain sedatives, or simply by aging, the sequence loses its timing, and the nightly transfer becomes less effective. Some sedative medications, including certain benzodiazepines and non-benzodiazepine sleep aids, produce sleep that appears deep on behavioral measures but suppresses N3 on EEG recording. These medications increase total sleep time and reduce awakenings. They do not reliably increase slow wave amplitude or duration. In some cases, they suppress the delta oscillations that define N3, producing a pharmacological state that resembles sleep without providing its restorative architecture. This is not a reason to avoid medication for sleep disorders. The clinical decision is complex and individualized, but it helps explain why some people who take sleep medication still do not feel restored in the morning. The brainstem, the most ancient part of the brain, at the junction between the brain and spinal cord, contains the neurons that initiate and maintain deep sleep. Specific nuclei in the brainstem, including the ventrolateral preoptic area, release GABA, the brain's primary inhibitory neurotransmitter, in coordinated waves that quiet the arousal systems. The thalamus is silenced first. Then the cortex begins to synchronize. The process works bottom-up. Ancient systems initiate, and evolutionarily newer systems follow. A mouse's deep sleep and a human's deep sleep share the same molecular initiators, the same inhibitory chemistry, and the same slow-wave signature on EEG. The repair protocol is old. The liver and other peripheral organs maintain their own circadian rhythms, roughly tuned to the central clock in the brain, but capable of drifting if the timing of meals and activity diverges from the light-dark cycle. During N.3 sleep, the synchrony between central and peripheral clocks is reinforced. Hormonal signals, particularly the growth hormone surge and the cortisol nadir, carry timing information to peripheral tissues, helping them calibrate their own repair cycles to the same schedule. Chronically disrupted sleep gradually desynchronizes these peripheral rhythms from the central clock. The consequences: metabolic disorder, impaired organ function, disrupted digestion, emerge slowly and are rarely attributed to their origin. The olfactory system, the sense of smell, has an unusual relationship to sleep. During deep sleep, the olfactory bulb continues processing some chemical signals from the environment, even though most other sensory pathways are gated by the thalamus. Researchers have used this property therapeutically. Low concentrations of pleasant scents delivered during N3 sleep have been shown, in some studies, to enhance memory consolidation, particularly for emotional material. The olfactory pathway bypasses the thalamic gate and runs directly to the hippocampus, where memory formation occurs. It is one of the few sensory channels that remains partially open during the brain's deepest rest. The skin undergoes repair during deep sleep, regulated by the same growth hormone surge that drives muscle and organ restoration. Collagen synthesis accelerates during N3. The barrier function of skin, its ability to retain moisture and exclude pathogens, is restored at the cellular level overnight. Skin is the body's largest immune organ. Its overnight maintenance involves the replacement of surface cells, the repair of damage from ultraviolet exposure and environmental stress, and the recalibration of inflammatory signaling in dermal tissue. Chronically poor N3 sleep is associated with increased skin sensitivity, slower wound healing, and exacerbated inflammatory skin conditions. The phrase beauty sleep is anatomically precise. Sodium and potassium balance across neuronal membranes. The ionic gradient that allows neurons to fire is maintained during sleep through a process called ionic homeostasis. During waking, neural firing gradually depletes the potassium-sodium gradient across millions of cells. This contributes to the subjective experience of mental fatigue. The signal quality of the neural network literally degrades as the day progresses. During N3 sleep, ion pumps in neuronal membranes restore the gradient cell by cell. The neurons of tomorrow's waking brain are being recharged tonight. This is not metaphor. The electrical potential across each cell membrane is being reset by molecular pumps running in the dark. The hypothalamus, which orchestrates deep sleep onset, also manages the body's inflammatory regulation. During N3, certain pro-inflammatory cytokines, chemical messengers that promote immune activity and tissue repair, are elevated. This is intentional. The immune system uses the quiet of deep sleep as a window for maintenance, detecting and responding to cellular damage, clearing dead cells, and preparing tissues for the demands of the following day. The elevated inflammation of deep sleep is controlled and purposeful, very different from the chronic low-grade inflammation associated with poor sleep. The body has allocated the inflammatory response to the sleep period, the only time it can proceed without competing with waking demands. The brain's blood flow pattern changes during deep sleep in ways that appear to actively serve the glymphatic clearance process. During N3, arterial pulsations in the brain, the rhythmic pressure waves produced by each heartbeat, synchronize with the slow oscillations of delta sleep in a way that drives the flow of interstitial fluid. The heartbeat becomes, in effect, a pump for the glymphatic system. This coupling between cardiac rhythm and brainwave rhythm is an area of active research. Early findings suggest that cardiovascular health, particularly arterial flexibility and heart rate variability, directly impacts the efficiency of brain fluid clearance during sleep. Auditory tone therapy during sleep. The technique of playing brief, quiet tones timed to the rising phase of the slow oscillation has shown consistent effects on slow wave amplitude in multiple controlled trials in adults. The technology works by using a monitoring device to detect the brain's own delta rhythm and playing a tone at the precise phase that reinforces the wave. The result is a measurable increase in slow wave activity without disrupting sleep continuity. Cognitive benefits. particularly in declarative memory, have been documented in the morning following enhanced slow-wave nights. Commercial versions of this technology are now beginning to appear, though research-grade precision has not yet been fully replicated in consumer devices. The hippocampus, which holds the day's experiences in temporary storage before transferring them to the cortex during deep sleep, is sensitive to cortisol. High cortisol impairs hippocampal function, reducing the capacity to form new memories during waking and disrupting the replay process during sleep. This creates one of the more self-reinforcing patterns in sleep biology. Stress elevates cortisol. Elevated cortisol impairs deep sleep. Impaired deep sleep leaves emotional memories incompletely processed. Incompletely processed emotional memories increase baseline stress. Understanding this loop and the biological specificity of where it runs is part of what makes the neuroscience of sleep directly relevant to anyone navigating a period of sustained difficulty. The body's pH regulation, the acid-base balance that maintains the precise chemical environment neurons require, is managed, in part, during sleep. Metabolic activity during waking produces acids as byproducts. The kidney and lungs compensate continuously, but their efficiency decreases with fatigue and stress. During deep sleep, metabolic rate falls, acid production decreases, and the kidney has time to excrete the accumulated acid load at its own pace. Blood pH moves toward its optimal baseline. People with chronic kidney disease or respiratory conditions often have disturbed sleep, partly because the pH management that sleep provides is precisely what those conditions compromise. The research on deep sleep and longevity is still maturing, but the direction of evidence is consistent. People who maintain healthy sleep architecture into older age, including preserved N3 amplitude and duration, show slower rates of cognitive aging, lower inflammatory markers, better metabolic health, and greater cardiovascular resilience. The association is robust enough that sleep architecture is now being studied as a potential biomarker for biological aging. A measurable signal that may capture more information about how the body is doing than chronological age alone. The circadian rhythm of deep sleep, its concentration in the first half of the night, creates an asymmetry in how sleep loss affects the body. Losing the early hours truncates the N3 window when it is largest and most productive. Losing the late hours truncates the REM period, which tends to dominate the second half. Both losses have consequences, but they fall on different systems. The person who goes to bed late and wakes on schedule is likely losing N3. The person who wakes too early and cannot return to sleep is likely losing REM. Both feel tired. The origin of the tiredness and the repair that did not complete is different. During the day, the brain generates electroencephalographic patterns dominated by high frequency, low amplitude waves, beta and gamma waves in the range of 12 to 40 Hz. These fast patterns reflect the fragmented, parallel processing of waking thought, many circuits active simultaneously, many things being held at once. Delta sleep inverts this completely. The waves are low frequency, high amplitude, and synchronized. The brain moves from doing many things quickly to doing one thing slowly. This is the shift that makes restoration possible. The fast brain cannot repair itself. It is too occupied. The slow brain can. The transition between them is the most significant state change the human nervous system undergoes in any 24-hour period. The experience of N3 sleep, if it could be experienced, would feel like nothing at all. There is no dreaming in deep sleep. The brain regions that generate narrative, imagery, and the sense of continuous experience are offline. There is no body awareness. Proprioception is suppressed. There is no sense of self, the default mode network, which maintains the sense of being a continuous self across time, is among the most quiet structures in the brain during slow-wave sleep. What is happening is immensely active. What it feels like, from the inside, is absolute absence. Deep sleep leaves no trace. It cannot be remembered. It can only be felt in its aftermath, in how the waking self arrived. The interplay between sleep spindles and slow oscillations in N3 has been linked to individual differences in general intelligence and learning speed. People who generate more sleep spindles per hour of deep sleep, and whose spindles are better synchronized with the slow oscillation rhythm, tend to perform better on tests of fluid intelligence, learning acquisition, and cognitive flexibility. This is not because intelligent people sleep more. It is because the efficiency of the slow oscillation spindle coupling predicts how much new information is transferred and integrated each night. Intelligence, in part, may be a function of how well the brain runs its nightly update. The glial cells that shrink during N3 to expand the brain's drainage space, called astrocytes, are also involved in regulating synaptic transmission during waking. The same cells that manage the extracellular environment during deep sleep are the ones that regulate the space between neurons during waking thought. The quality of your thinking during the day is partly a function of how well those cells were able to perform their overnight maintenance. Rested astrocytes maintain tighter control over the extracellular chemistry that allows neurons to communicate precisely. Fatigued astrocytes, after shortened or disrupted deep sleep, show reduced efficiency in this regulatory role. Shift work, working nights on a schedule that inverts or disrupts the normal sleep-wake cycle, is associated with measurable reductions in N3 amplitude and duration, even after years of adaptation. The circadian clock does not fully adjust to an inverted schedule, because light exposure continues to signal daytime even when the body is trying to sleep. The deep sleep that shift workers achieve is lighter, shorter in slow-wave content, and less restorative on most biological measures. This is one reason shift work is associated with elevated rates of metabolic disease, cardiovascular events, and cognitive decline, effects that reflect the ongoing suppression of the sleep stage that most of the body's maintenance relies on. The relationship between exercise and slow-wave sleep offers one of the most direct levers available for improving deep sleep quality in adults. Beyond the adenosine and thermoregulatory effects, exercise increases the production of brain-derived neurotrophic factor, or BDNF, a protein that supports neuronal growth and synaptic plasticity, and is associated with deeper, more restorative sleep. BDNF levels rise after aerobic exercise and remain elevated overnight. They are also elevated during and after N3 sleep, suggesting a bidirectional reinforcement exercise improves deep sleep. And deep sleep provides the neurochemical conditions that make exercise more effective the following day. Dietary patterns influence sleep architecture in ways that are still being mapped, but some consistent findings have emerged. Diets high in refined carbohydrates and added sugars are associated with more fragmented sleep and less slow-wave activity. Diets higher in fiber are associated with more N3 sleep on objective measurement. The mechanism may involve the gut-brain axis. Gut microbiota composition affects the production of neurotransmitter precursors, including serotonin and GABA, which influence the brain's sleep-promoting circuitry. A significant proportion of serotonin, a precursor to melatonin, is produced in the gut, not the brain. What the gut does during the night is connected to what the brain does. At this point in the night, the intellectual answer to tonight's question is clear. The brain's deepest sleep stage is not a passive rest. It is a maintenance protocol, metabolic, immunological, neurological, cardiovascular, that the brain initiates by generating coordinated, synchronized delta waves. And that runs on a biological schedule that cannot be moved by intention alone. Deep sleep is not more sleep. It is a different state the brain enters to do what waking life cannot. The remaining facts carry a different quality now. They are not adding information. They are letting the information land where it needs to. Breathe slowly now. Your heart has been moving without interruption through every fact of this night. Not once asking permission. Not once slowing for the words. The delta waves are already present in you. Not because you are trying, because the body knows what to do when the thinking finally quiets. The slow oscillation is the brain's oldest rhythm. It does not need to be earned. It only needs enough stillness to begin. Let the weight of where you are be enough. The facts that remain will arrive more softly than the ones before them. We're almost there now. The brain in deep sleep is running a protocol so old it predates language. Every mammal that has ever lived has entered slow-wave sleep. Every one has had the same glial shrinkage, the same cerebrospinal surge, the same downstate silence and upstate synchrony, what we call rest. Biology calls maintenance, what we call sleep. The nervous system calls the only window for doing what must be done. A single night of deep, complete slow-wave sleep does not undo weeks of accumulated deficit, but it resets the trajectory. The morning after genuine N3 sleep, the prefrontal cortex is sharper, the emotional baseline is steadier, the pain threshold is higher, the immune system has updated. The body begins the day from a different starting point, not a perfect one, but a better one than yesterday. Melatonin, the hormone most commonly associated with sleep, does not produce deep sleep directly. It signals timing. It tells the brain that darkness has arrived, that the circadian window is open, that the conditions for sleep are present. What happens inside that window? Whether N3 develops, whether it runs long enough, depends on sleep pressure, temperature, stress hormones, and the integrity of the sleep-promoting neural circuits. Melatonin opens the door. The brain decides whether to walk through it. The slow oscillation of N3 travels across the brain at a speed of roughly 3 feet per second, about the speed of a gentle wave spreading across still water. It begins at the frontal pole and rolls backward, involving more and more of the cortex as it moves. This wave is not metaphorical. It is measurable, directional, and carries a distinct neurochemical signature as it passes through each region. The brain in deep sleep is, moment by moment, washing itself in waves. When the glymphatic system clears the byproducts of a day's neural activity, The cerebrospinal fluid carries these molecules to the bloodstream, where they are processed by the liver. The brain's metabolic waste goes to the liver. The liver's processing goes on through the night. The organs are collaborating on a maintenance schedule, the brain coordinated. One system clears, another processes. Neither waits for morning. Researchers have found that people who consistently achieve good quality slow-wave sleep, even as they age, show measurably slower biological aging in their brain tissue, the telomeres in their neurons, the protective caps on chromosomal DNA, degrade more slowly. The accumulation of age-related cellular damage is reduced. Deep sleep is not a cure for aging, but it is one of the processes through which the rate of aging is negotiated. Night by night. There is no substitute for sleep in the repair protocol it runs. Caffeine blocks the feeling of tiredness. Rest reduces metabolic demand. Nutrition provides the materials for repair. But none of these processes replicate the specific, sequenced biological work of slow-wave sleep. The brain can be kept alive without it. The brain cannot fully maintain itself without it. This distinction between functioning and maintaining is where the nightly importance of deep sleep lives. The silence of deep sleep, the absence of experience, the absolute blankness, is a protective state. The brain is not accessible during N3. No experience is being formed that could be disrupted by emotion or colored by the anxieties of the day. The material of the day is being processed, but not by the conscious self. It is being processed by the system that preceded the self. That older system does not worry. It works. Growth hormone released during N3 sleep does more than rebuild muscle. It influences the repair of connective tissue, tendons, ligaments, the dense collagen structures that hold the body together. Athletes who prioritize sleep show faster recovery from soft tissue injury. The same mechanism maintains the integrity of joints and connective structures in non-athletes as well. The nightly growth hormone surge is one of the reasons physical health, in the long term, cannot be separated from sleep health. The body's circadian system has a preference for synchrony, for the light of morning, the dark of evening, the temperature of night arriving in predictable sequence. When this sequence is consistent, the brain's sleep machinery runs more smoothly. When it is irregular, variable bedtimes, varying light exposure, shifting meal times, the circadian clock loses its precision, and the signal that drives N3 becomes less crisp. The brain can still reach deep sleep. It just has to work harder to get there. Every night, the brain makes a decision about how deep to sleep. The decision is made in biology, not in intention, but the conditions for the decision are shaped by the hours before sleep. The light exposure, the timing of the last meal, the temperature of the room, the level of stress that followed you to bed. These conditions are not destiny. They are inputs. And inputs can be changed. The sound environment during deep sleep influences its quality more than most people realize. Continuous low-level noise, traffic, appliances, urban sound produces micro-arousals that show up on EEG as interruptions to slow-wave activity. A quiet room does not guarantee deeper sleep, but a noisy one reliably fragments it. Sound that crosses the arousal threshold pulls the brain out of delta, even briefly. And the slow-wave episode must begin again from a lighter state. The skin temperature of the hands and feet rises during the transition to deep sleep, as blood vessels dilate and the body radiates heat from its periphery. This rise in skin temperature at the extremities is one of the most reliable physiological precursors to N3 onset. Warming the feet. Through warm socks or a warm water bottle at the foot of the bed has been shown to accelerate this process and reduce sleep onset time. The body is trying to shed heat from its surface. Helping it do so is, in a literal sense, helping the brain get to work sooner. The relationship between hunger and deep sleep is reciprocal. Hunger activates the hypothalamus in ways that compete with sleep initiation. A small amount of complex carbohydrate before bed can raise serotonin, a precursor to melatonin, and reduce the alerting signal of mild hunger, without producing the metabolic disruption of a large late meal. The goal is not to go to bed full. It is to go to bed without the physiological signal that tells the brain the body needs to seek food. That signal and the signal for sleep originate in the same region. They cannot both run well at the same time. The slow wave of N3 sleep is one of the most orderly electrical events in the human brain. It is more synchronized than most waking activity, more regular than dreaming, more patterned than deep relaxation, which produces alpha waves. Faster, smaller, less coordinated. Nothing the waking brain does produces the same breadth and amplitude of coordinated neural activity as the delta wave of deep sleep. It is the most thoroughly organized state the brain enters. It is called slow. It is electrically precise. The morning after a night with intact slow-wave sleep, the brain's default mode network, quieted during N3, reactivates with greater coherence. The connections within it are cleaner, more regulated. This correlates with greater self-awareness, more flexible thinking, and reduced rumination throughout the following day. Deep sleep does not only repair the body, it restores the architecture of the mind that you will wake up inside. There are people who are brief sleepers by genetic variation. Individuals with a mutation in a gene called ADRB, one who function optimally on 6 hours or less. This mutation is extremely rare, affecting far less than 1% of the population. Most people who believe they function well on 6 hours have adapted to chronic partial deprivation and are no longer aware of the baseline from which they have drifted. The adaptation is real. The functioning that returns after consistently complete sleep is often a revelation. The brainstem structures that initiate slow-wave sleep are also responsible for reducing muscle tone throughout the body during N3. Skeletal muscles become nearly atonic, deeply relaxed in a way that is not possible during waking. This relaxation is a biological requirement. The brain needs the body to be still so that the resources devoted to motor maintenance can be redirected to the internal work of the repair protocol. Stillness during deep sleep is productive. The absence of movement is the condition for repair. The heart, during N3 sleep, beats in a pattern of low frequency. High amplitude heart rate variability. The intervals between beats are longer and more variable, a sign of parasympathetic nervous system dominance. Cardiologists recognize this as the signature of a cardiovascular system at rest in the deepest sense, lower vascular resistance, lower myocardial demand, lower blood pressure. The deep sleep the heart gets from each full N3 episode is among the most cardiovascularly restorative periods in the 24-hour cycle. The heart does not rest between beats. It rests in the valleys of sleep. One of the most consistent predictors of how much deep sleep a person will get on any given night is how much physical and cognitive work they did during the preceding day. The brain and body respond to the demand of the day with a proportionally deeper sleep drive. Rest days and low-demand evenings are associated with lighter, slow-wave sleep. This is not a problem. It is a calibration. The system provides what the system needs. On the nights that matter most, after the days that cost the most, the brain tends to reach deepest. The language of the body during deep sleep is the language of repair, slow, internal, quiet. It is the opposite of the language of effort. There is nothing to do in N3 sleep. No choice to make. No attention to maintain. The brain has taken over completely. The body already knows what to do with the night. It has been doing it, without instruction. Since before you were born, the breath slows, during N3, to its lowest rate of the night, between 8 and 12 breaths per minute, slower and more regular than the breathing of waking. Slower even than the breathing of light sleep. This slow, even rhythm assists the glymphatic flow and reflects the depth of autonomic calm. When the breath slows to this rate without effort, the brain is in the window. The window is open. The work is running. Every night you have ever slept, your brain has entered slow-wave sleep, or tried to. On the difficult nights, it still attempted the protocol, still generated whatever delta it could in the time available. The brain does not give up on repair because the conditions were imperfect. The drive to reach deep sleep is among the oldest biological imperatives in the mammalian body. More persistent than appetite, more reliable than temperature regulation. The brain will always move toward its deepest sleep. It only needs enough of the night to get there. In the final facts of this night, the question that opened it has been carried through 100 layers of answer. What does the brain actually do in the hour most people never reach? It clears. It consolidates. It recalibrates. It repairs the tissue, the chemistry, the electrical potential, the ionic balance, the immune architecture, the hormonal cascade. It runs the oldest maintenance protocol in the mammalian body in the one window. The slow, dark, coordinated window that evolution set aside for exactly this. The answer to tonight's question is not a finding, it is a reorientation. Deep sleep is not the absence of consciousness. It is the presence of something older than consciousness. A biological intelligence that does not require your awareness to do its work. You do not reach deep sleep. You allow it. You create the conditions. And the brain takes you there. What happens next is not yours to observe. It is yours to wake up inside of. Lighter, cleaner, a little closer to whole. Deep sleep is not more sleep. It is what waking is for. The slow wave was always the destination, not the drift of dozing, not the shimmer of dreams, but the place where the brain finally moves at the pace of its own repair. We followed the delta from its first quiet appearance tonight, through the glymphatic surge, the hormone cascade, the hippocampal replay, the synaptic pruning, to here, where the protocol closes. What began as a question about the hour most people never reach became something closer to a recognition the brain has been doing this work for every night of your life. Unobserved, unhurried, ungiven credit. The tiredness you woke with was not failure. It was a signal about where the night could not go. And tonight, it went there. The Relaxing Facts About Sleep playlist has been tracing the science of rest one mechanism at a time. The next video follows what happens when the night does its work. And the waking brain carries something forward that sleep built. That video is waiting whenever you are ready. Something is still now. The way the brain goes when the slow wave finally arrives. Let your body give itself over to the quiet that is already here. If sleep comes, let it carry you into the hours you won't remember, the ones doing the work you came here for. But if your eyes are closed, stay there. Rest well, weary one. The night always knew what to do with you. Where science becomes the softest place to rest. Good night.